All right. Well, thank you, Susan. So it's a pleasure to introduce Stephen, who did his bachelor's of science at the University of Ottawa in major in chemistry, and also did a master's there. I think actually mostly in genetics, looking at hypertension, and so got kind of interested in research and came here to do epidemiology, more population-based research. And so he's going to talk to us about his PhD project. Stephen's also been heavily involved with EBOS, the Student Association within the Department of Epi, having been on the executive. And he actually has some clinical experience in that he resuscitated one of his hockey mates 2 years ago, and a 45-minute out of hospital cardiac arrest, and his colleague survived with neurologically intact. So this is really quite an amazing feat in terms of out-of-hospital resuscitations. However, today he's going to talk to us about more in-hospital events. So Stephen, I'll turn it over to you.
Thanks, Jay, and thanks for reminding me of that very, eye-opening moment, and it was kind of amazing to kind of be so close to the person that experienced such an event. But I'm glad that he is doing well, and I can confirm that he is completing his studies to today, and very grateful for the team that worked with him at the MUHC, which actually included Dr. Brophy as his physician that I got to meet when I went to visit him. So it's kind of a small world. But I want to welcome and thank you all for joining me today. You'll notice that if you can't hear me very well, I've included closed captioning to hopefully help clarify any of my mumblings as I talk too quickly throughout this presentation. And I'd like to start the presentation out by confirming that I'm here live and I'm not a cat. So for any of you who are enjoying the little brief social media kind of comedy that's going around, if you haven't, please go check it out. But I'll be describing my thesis projects that are involved in the investigation of dual antiplatelet therapies in patients with acute coronary syndromes. So a quick overview of what I'm going to talk about today. We have the background of what acute coronary syndrome is, a bit about dual antiplatelet therapies, the different options, some of the drawbacks or potential limitations, my research objectives, and then overview of all my 3 thesis projects. And then talk about the overall impact of what my research will hopefully do in the context of antiplatelet therapy treatment of ACS. So acute coronary syndrome is a term that describes a group of conditions that are characterized by a sudden reduction or loss of blood flow to the heart muscle, comprising of unstable angina, non-ST and ST elevated myocardial infarctions. You can see here there's some narrowing, and there's a blood clot that can get blocked here and then prevent blood flow down below tissues. And obviously this impeded downstream blood flow can impede then nutrients and waste being removed and brought to these muscle tissue, and then that can result in muscle necrosis. And that's obviously an issue for a major organ that is used constantly. Oops. So in the context of Quebec, there's been some major advancements in cardiovascular medicine in the past several decades. We can see that there's been a drop in cardiovascular related death rates from 1975 and towards today. So we've gone from over 800 in men and 500 events per 100,000 persons down towards 200 and 100 and closer to today's date. And you can also see here that this reduction continues, albeit much slower in the more recent years. And now cardiovascular mortality has been surpassed by cancers. That should be guessed. Cancers have probably not dropped as much, but just that a decline in cardiovascular outcomes has been more rapid. So it is not the #1 cause of death in Canada anymore, which is fortunate. But with that successful reduction in cardiovascular mortality has come with an increased prevalence of those who survive an ACS event. So you can see here that the prevalence is kind of plateaued around 10% for males and around 6% for females of those who have or are living with an ACS event in our Quebec population. And it's important because prior ACS patients, or ones who've had an ACS event, are at a higher risk of a recurring ACS event and are then a leading cause of hospitalization, and so a burden to our healthcare system. And so preventing a recurrent cardiac event after your first one is an important goal in managing ACS patients. And so antiplatelet therapies come into play and are a key in preventing the blood clotting that we saw in the previous slide that can prevent blood flow through heart arteries. And aspirin was one of the first identified platelet adhesion antagonists in the 1980s, and it remains a crucial component of the treatment guidelines to today. But dual antiplatelet therapy means it's the concomitant use of aspirin plus a second antiplatelet drug. So there's two different drugs combined in now your treatment. So one of the options is Clopidogrel. So Clopidogrel is another platelet cell receptor inhibitor and prevents aggregation, but on a slightly different pathway on the platelet. And multiple RCTs demonstrated that the combination use, so the use of aspirin and Clopidogrel together, this was associated with a reduction in cardiovascular rated outcomes when compared to aspirin or to Clopidogrel alone. And I'll probably call that DAPC quite often. Hopefully I try and stay somewhat stable in terms of my usage. And then DAPC became the foundation of treatment following the addition of Clopidogrel. But Clopidogrel is a prodrug, so it requires liver activation, or so enzymatic activity in the liver to convert it to an active form in its platelet adhesion antagonism. And so there's a proportion of the population who do not have the liver enzymes or enough to convert it as quickly. And it's kind of similar to, you may have heard, people who don't digest alcohol. So that's kind of the same idea, is that you don't have the same enzyme distribution within your body to break it down and be as effective. So there's a proportion of the population that might not be as reactive to Clopidogrel. And so it might be a little less efficient. And so then they developed a new one. So Prasugrel, it's also a prodrug, but only requires a one-step activation. And it is considered to be a slightly improved or quicker onset compared to Clopidogrel. So Prasugrel was developed after Clopidogrel as an improvement. And then lastly, we have the third one, which is Ticagrelor, which is is a active direct acting drug, and it does not require any liver activation and is considered a quicker onset than both Clopidogrel and Prasugrel. And you can see here now the progression from the different drugs from Clopidogrel, Prasugrel to Tycagular in terms of the three different treatment options. So now we have three options. You have an ACS patient. Which one do you choose then? Well, let's see what the guidelines say. Canadian guidelines recommends Tycagular and Prasugrel over Clopidogrel. And based on what we saw before, it might make sense because Tycagular might be able to be managed better across the population. However, if you look at the evidence cited in the Canadian guidelines, it's a study looking at Prasugrel or Clopidogrel versus a placebo. So it wasn't randomized to either of these two drugs. It was randomized to placebo or one of the two. And then they look at a trial looking at two different dosages of Tycagular versus aspirin. So I personally think this is just a clerical error. So I chalk this up to just them misciting evidence in their guidelines. And so I kind of have to point that out, and I base that off of the other two. So I'll kind of just keep that in mind here. So the European guidelines, same as the Canadian ones, recommends Tycagular and Prasugrel over Clopidogrel. And they cite these three RCTs, these multi-center RCTs, comparing Prasugrel Clopidogrel, Prasugrel Clopidogrel, and then Tycagular Clopidogrel. And you'll see these three studies in a bit. And then lastly, the American guidelines talk about, does not explicitly recommend a dual antiplatelet therapy or one or the others. And it cites evidence of using all three, and it uses these two, but it does actually within it have an interesting quote that isn't in their flow diagrams by any means, but it says it is, quote, reasonable to use Ticagrelor in preference to Clopidogrel, end quote. So let's look a little bit closer at the evidence and see where maybe they got these ideas from. So as you see here, it's important to highlight that there's an efficacy component and a safety component. efficacy being the prevention of a recurring ACS event, and you're reducing the ability of your blood to clot, and a safety component being bleeding because you're reducing your clotting ability, and you obviously don't want to have bleeding is bad as well. So you have to kind of pay attention to both of these. So balance these benefits. And we see here that from the Triton and Trilogy trials, the efficacy for prasugrel are shown to be beneficial for coronary outcomes when compared to clopidogrel, but they have a higher safety outcome, or sorry, a higher safety risk. So something that needs to be balanced when you look at these drugs. However, if you look a little more closely, I found this on the 29th page of the supplement in the Triton trial, and this is the information on safety. So this is basically their safety outcome, but stratified by different region. And so you can see there's a bit of heterogeneity. And now my question, I guess, is to those of you who are more familiar with meta-analyses, do you trust that this estimate is captured from this? And I'm not saying you should have this answer right for me right now. You probably have some ideas of how you could go about do that. But let me add some more information to that. If you did a study in over 50 countries like Triton was done, I would personally at least expect some level of regional variation in the efficacy and safety outcomes across those 50 enrolling countries, be it because they have different medical systems, different treatment capabilities, the health of their general population, different cultural practices, dietary differences. So there's a range of things that I think could impede, or not impede, but provide some variation in terms of the effective treatment in different regions of the world. So if you, now I guess I wonder, did they account for this heterogeneity in this estimate? I personally was unable to find anything saying that they did account for this heterogeneity, and I was unable to reproduce this result from this data because it wasn't rich enough for me to pull it out. So I guess in summary, the dual antiplatelet therapies, DAT-P here shows a benefit for efficacy, but a potential risk and safety. And there may be an underestimation of this variance based on their limited use of the heterogeneity between the different study regions. So let's move on to Ticagrelor versus Clopidogrel. So there's the Plato study, and it was done again in a large multi-center RCT, done in over 40 countries. I think it was 42. And they have over 18,000 patients. So a lot of evidence, which is good for us. And the efficacy endpoint that they found for was a 0.84, so benefit compared to clopidogrel, significant, and then a safety endpoint that was around 1.04 and non-significant. So I had a better efficacy profile and similar safety profile to clopidogrel, which is good. But similar to the previous slide I showed you, there is some regional variations. And the FDA reported in their safety documents, while looking through this, that there needed to be more research on the regional heterogeneity that was not explained in the original study. And you'll notice here that the US group has a higher hazard ratio of 1.25, non-significant, because they only had about 1,000 or just under 2,000 patients. And so there was a signal here that was a bit concerning. And so personally, it depends on the individual. I'm more of a random effects at all times kind of person, but some people might disagree with that, and I'm totally okay with that. But the only thing I'd like to highlight from this is that if you use a hierarchical or a random effects model, you can see that the confidence intervals grow a bit and take into account some heterogeneity that comes from these different studies. And so, but just by selecting a random effects versus a fixed effects model, you can see that your interpretation of this trial outcome would change. And whether or not you agree which one is correct, I think you would agree that this would be a bit concerning in terms of your overall robustness of this estimate. So are we comfortable with these large multiple central trials just simply pool this data, or do you think that we should be describing more variations about these estimates and have more focused estimations in our populations? So again, regional variations are not accounted for in the analyses, not described. It seems like we're underestimating the variance of the outcomes, and we have some North American subgroup signals that are kind of a bit concerning. And I think we should do more research into these populations as the FDA suggested. So it comes to the purpose of my entire thesis and my PhD now, and it is to investigate these uncertainties around dual-antiplatelet therapies and do a comprehensive investigation to the association of DAP regimes and cardiovascular efficacy and safety outcomes in patients that have an ACS event. So how am I going to do this? I have 3 projects that I'll go through a bit quickly, or I'll try and go through in some detail here. The first being a Bayesian network meta-analysis, a second one, an observational study using RAMQ data, and then the third one, a TC4 pragmatic trial, which personally I think is the most interesting one because I'm sure a lot of you have seen these other study designs. And I apologize to any of you who are here who've already heard this talk. I know I have some colleagues who get to listen to this a bit too often, but you get to hear it again. So, and I haven't seen you in a while, so this is exciting. So before we get too far into the details of the research, because I do some Bayesian stuff, I just wanted to give a brief overview to help remind people of Bayesian statistics or bring some people onto the same page just on the surface level if you haven't seen it before. But basically, most research is motivated by some prior information or information that comes before you design your study. So you have some reason to engage and implement your study. And it seems logical only to use that data instead of ignoring it. So Bayesian statistics involves using that prior information and combining it with the likelihood of the data you collect into a posterior, or your results, your final output. So this would be your results from your prior and the likelihood of your data. This is just for illustrative purposes here. This is not any specific data. I just created this, by the way. So you can simulate this posterior distribution by Monte Carlo Markov chain sampling, by sampling from the prior and likelihood joint distributions. So again, you have prior information from before the study, you collect the data from your study in the likelihood, and you combine them into the posterior. And then what's great about this is that this posterior now can become the prior to your next study. And so you can continually update your information as you collect more data. And so it's kind of the natural research process here. Okay, so into the first objective of the Bayesian network meta-analysis. So for those not familiar with a network meta-analysis, it's also known as a multiple treatment comparison. It's a technique that just allows for synthesizing effect measures from more than 2 exposures. So you're probably more familiar with the pairwise meta-analysis where you, for example, have Clopidogrel versus prasugrel and you can combine all the studies that compare those 2. Or now, because I have 3 exposures, now you can give them each a node and then compare across these different nodes. And so it just allows for more flexibility in your meta-analysis. So here's a bit closer on my structure. This is just kind of a visual demonstration here. But the solid lines are what we call direct effects. So you can think of Plato comparing Clopidogrel to Tycagular as this black line. And you can think of Triton comparing Clopidogrel to prasugrel as this dark line. But then you can also calculate indirect effects. So you take those direct effects from prasugrel and Clopidogrel, subtract them. Well, in this case, I use prasugrel and Tycagular, and you can calculate the dotted line here, which would be in this one, in this case. And that's your indirect effect. So you're gaining a bit more information than if you just did a head-to-head comparison of just 2 nodes. So you can now collect a bit more info. So a bit into the methods. I won't go too much detail because I think everyone's kind of familiar if you are with meta-analyses, and I don't think they lead to a very interesting content. And I know a lot of you probably spent plenty of time on Zoom for these meetings before. But essentially, I'm going to follow the Prisma network meta-analysis guidelines. There are specific ones for network meta-analyses for reporting. And I included a medical librarian to help me develop a search strategy for MBase, Medline Central, and clinicaltrials.gov. And then I have some conclusion criteria here, such as having an ACS diagnosis, being assigned to one of the therapies of interest, being an RCT, and then reporting at least on the efficacy outcome from the that I'm interested in. And they also, I'm also going to be doing quality assessment and data extraction using the risk of bias 2 assessment tool, but by 2 independent reviewers. So my analysis is going to involve, because I don't know if everyone's going to report the same way, I decided to choose or prepare a Bayesian hierarchical generalized linear model with a logic transformation, so a binary outcome, just in case I don't get survival reporting only, and I'll include a log follow-up time component to help account for some of that measurement of time. And essentially the hierarchical model will just assume that your effect measure comes from a distribution which will have a mean and standard deviation. And I can choose the best model using deviance information criterion to assess model fit. Essentially just similar to AIC or BIC for those, but specific to Bayesian network meta-analyses. My outcomes of interest. So this is going to be a theme across my 3 projects. But essentially the efficacy one is major adverse cardiovascular events, MACE, which is a composite of non-fatal MI, ischemic stroke, and death. And then I have a safety outcome, which is going to be a composite of significant or major bleeding. So a composite of major reported bleeding events in the studies. And then I've also planned some subgroup analyses here, which is looking at a North American population. I'm hoping that they'll report the data on them. And then I can also look at revascularization procedures, which is common in ACS patients. So some assumptions that go into your network meta-analysis here that should pay attention to: heterogeneity, which many of you will be familiar with as the within variation of pairwise contrast, and is commonly described using the I-squared statistic, standard in your standard meta-analysis. And then you have transitivity, which is the similarity between the pairwise contrast or across different arms of the network. So for example, you want to compare these studies to the studies in these comparing these nodes, and you do it more qualitative to make sure that they have similar population characteristics across them. And then lastly, we have this consistency assumption, which is comparing the direction and having sort of concordance in the direction of your direct effects as well as your indirect effects. You want to make sure that these 2 estimates that you get from the different evaluations are in the same direction. So where I'm at with this study. So in August, I ran the search strategy because I was waiting for a couple of trials that I knew were going to be published. And I identified over 10,000 articles. I duplicated them down to about 6,000 articles a few months ago. And I'm currently still in the screening process with a colleague that I had convinced to go through this with me, which is fun for them. So hoping I have this done in the next couple months and have some more information. So if you talk to me, hopefully in the hallways at some point, we can have some more better discussions about this. But I don't have any guarantee that I'm going to be able to pull North American evidence from this study. And with the limited North American evidence in the original Plato and Triton trials, they had only around 10 to 20% North American patients. My next objectives are going to focus on the North American population. So my second objective, the nested case control observational design. So it's going to leverage the RAMQ healthcare databases as well as ENSPQ, MSSS, and we're going to combine them using de-identified codes for individual patients, and I can get hospitalized admissions, diagnostic information, prescription drug, dosage, duration, doctor visits, specialty of doctor they visited, as well as 3-digit postal code and deprivation index as socioeconomic proxies for some information. And I'll be using a new user definition for dual antiplatelet therapy exposure. And what I mean by that is that in order to be in the cohort, you have to be in our database for 1 year to assess that exposure prior to your index event or your ACS event for us to assess baseline characteristics as well. And at that first ACS event will be our time 0 to assess exposure status. So when the moment you leave the hospital, you will have your time 0 and we will start assessing your exposure from there. And your exposure will start from the day of prescription. And because dual antiplatheters have a relatively short half-life, we're going to use a 2-day grace period at the end. So if you have a 7-day prescription, for example, your exposure will be for 9 days in our database. And if you have adjacent or overlapping prescriptions, then you'll be considered continuous exposure. And you'll see we'll do some sensitivity analyses for the grace period we selected. And then the 4 groups that I'm going to plan to identify here are those who are doing Clopidogrel, Ticagrelor, Prasugrel, and other being unexposed if you're not on a prescription based on our definition, or if for some reason you've got a different antiplatelet med that isn't common in our population. So the outcomes. So the outcomes, again, are similar to previously, being MACE and major bleeding, and they'll be identified using ICD-10 codes in our data sets for non-fatal myocardial infarctions, ischemic stroke and death, and then our major bleeding being hemorrhagic stroke and gastrointestinal bleeding. So what will our cohort look like? So this is kind of a visualization of how it hypothetically could look. For example, 2011 here, even though our data set starts in 2010, you need 1 year within the data set to collect your baseline information and make sure you're not using a dual-antiplatelet theory prior to your index event. And then you can enter the cohort with an ACS event, and then we'll collect your prescription information, and it can be intermittent or time varying. And then either you get lost to follow-up because you left the data set, the end of the study, or you had an event. So each line here represents one single patient. And when you want to run the NACE case control, you align all the patients' time zeros up on the y-axis, and you get to follow them across the follow-up time here on the x-axis. So time zero is here if you're hospital discharged, and you can then follow a person when you find a second event, so either a death, another ACS event, or a stroke. That becomes your risk set, and we're going to sample 10 patients within this risk set and match on follow-up time, sex, and age using a caliper of 2 years. So my analysis, I'm going to have a matched cohort of these using those covariates I just mentioned. And so you have to use a multivariable conditional logistic regression to account for clustering for matching. And I'm going to be, and then from this conditional regression, I'll get odd ratios that are unbiased estimators of your hazard ratio. So in the analysis, I'll have to account for some, because I don't have any randomizations, I'm going to have to account for some confounders, so comorbidities such as hypertension, diabetes, some general health ones, and then I'm going to include the socio factors that are proxies that I can for socioeconomic status. So here's a list of the ones that I'm planning to include in my research. Again, I apologize for being repetitive here, but like previous studies, the primary outcome will be MACE. The second safety one will be a composite of hemorrhagic stroke and GI bleeds. And then I'm planning to do a sex stratified analysis because if you recall from the start of the presentation, males and females seem to have different rates of cardiovascular disease. So we wanted to investigate if there's any heterogeneity of the treatment between these different populations. There's also, we're going to try and look within the revascularizations. So whether or not someone had a PCI, so percutaneous coronary intervention, or CABG, for example, and we're going to explore that. And then lastly, the individual components of the composite outcome. So look at non-fatal MI by itself, stroke by itself, and death by itself. So where I'm at with this. So we have the data set. It's quite a large data set over several years. I found over 200,000 unique identifying codes for patients. I don't have anything immediately for you because I'm still in the process of going through this. But the interest part of this is that it should provide a substantial number of North American patients compared to what the trials had. If you remember, we had probably no more than 2,000 patients in either trial. And so you're looking at almost 100-fold increase in numbers here. And hopefully we can get this done before the RCT. The RCT was actually started before this, but we probably will be able to get the results from this before the RCT is concluded, which is in a good transition over to my third portion, which is my final thesis project, which is the TC4 trial, or the Tycagular compared to Clopidogrel in acute coronary syndromes, a pragmatic randomized controlled trial. So just to highlight there, we're looking only at Clopidogrel versus Ticagrelor here. So randomized controlled trials, they're often considered the gold standard of research for causal inference. Proper randomization can provide confidence that confounding will be minimal issue when interpreting effects measures. So if they're so great that the gold standard, there must be a reason why we're not using them all the time in comparative effectiveness research. Well, obviously RCTs have some barriers. So for example, selection bias. Participants recruited to RCTs often don't reflect the clinical population for which we want to estimate our effects within. And this is often called generalizability or external validity issues. Another example is successful randomization. In order for a randomized trial to be useful, it has to have successful randomization. And this can sometimes be challenging. If you've ever tried to do this, it's not as easy as it sounds in practice. It sounds easy to go up and just flip a coin, but it gets complicated, especially when you're in a setting where acute care and rapid urgent care is needed. Thinking of that original individual Jay talked about, where he had a cardiac event on the ice and I got to participate and revive him. You don't have time to flip a coin before that happens. So that's another barrier, is having successful randomization. And then thirdly, resources. So you can imagine those trials that did end up doing it in 50 countries, there was probably a substantial amount of resources that went into not only doing the randomization process at urgent care level, but also for the follow-up time necessary, 1 year of follow-up in these patients. So it requires a lot of effort to collect that data. So our trial, we don't have endless pharmaceutical funding. We are publicly funded by CIHR. So we propose some solutions to try and get around some of these barriers, especially the financial cost one. The first one is that all patients at the ACS patients at the MUHC are going to be eligible for our study. We're recruiting through the cath lab. And so if you come in to get a procedure and you're going to be assigned a treatment, you are now available for our study. For randomization, we're going to use a time cluster randomization design. I'll explain this a bit more on the next slide because it might not make sense in itself, but essentially we're randomizing you by periods of time. So depending on whatever month you arrive at the hospital, you get your exposure. And then follow-up done, instead of having someone call up every single patient, we're actually going to be leveraging the RAMQ data again to get our clinical outcomes, similar to what we did in the observational study. So here's kind of a visualization. So the first problem being generalizability. So you have all patients available. So our clinical population that arrives at the MUHC is eligible. Depending on what month you arrive at the hospital is when you get your exposure to Ticagrelor or Clopidogrel DAP therapy. So our trial started in October 2018. And so for those months of October and November, you were assigned to Clopidogrel. From December through to the end of January, you then got Tycagular. And this alternating exposure randomization continued up until today, as we are just starting to wrap up recruitment. And then we're going to follow these patients in each group using the RAMQ database for their outcomes, such as MACE and for efficacy, and then their bleeding for safety outcomes. So my analysis is going to be a time to event analysis, so survival, and it's going to be a hierarchy fatality model with random effects for the clustering. So again, it just means that your effect within the group is going to come from a distribution. And we're going to use an intention to treat analysis as the primary analysis. It's pretty standard for randomized controlled trials, where your assigned treatment at day 1 is the treatment we assume is you're going to have throughout the study. And I'm hoping we're going to be able to get some data on whether or not they adhere to the study drug or switch, and maybe implement time variant analysis too at some point, but I'm not sure if we're going to be able to collect that just yet. So my analyses are going to be my primary ones, similar to previous studies I described, MACE and safety as being bleeding events. And then I'm going to do my sex stratified subgroup analysis, as well as the individual outcome components that are part of the composite outcomes. So I'm going to go in a bit about Bayesian priors here for a second to hopefully help ground my next slide. So we said we can use prior data when doing a Bayesian analysis. So you want to combine your prior data with your likelihood. So choosing your prior is the challenge. How do you convince people which prior to select? So there's different options. You can choose, for example, this green dotted line, the gamma prior, which has a density up here, but is relatively flat across the entirety of the data frame. And you can see that the likelihood, the blue, is basically the same as the posterior. So essentially the prior becomes non-informative. You can also do this with a prior that is flat, so it's the same probability across the entire thing. And we can see here in this case, it provided the same result. The thing about the gamma that you have to pay attention to is that if your likelihood comes in a region that's close to this density here, it can really affect your posterior. So you can also choose a weak normal. We don't think that really extremes of your data are just as equal as the middle. So maybe you can add a weak normal where the middle is a little bit higher, but doesn't have a lot of information. And then of course you can choose what we call historical or skeptical, or historical optimistic priors, which are all contextual, but essentially you can choose different priors with the same likelihood and get a different posterior. So you can see here, I use the same likelihood, the blue but I selected a different prior for these two models and I get a slightly different posterior. So these are just illustrative purposes, but hopefully it helps you understand what we're doing for the trial here. So my range of Bayesian priors for the TC4 trial. So my historical skeptical priors, what I want to pick is going to be the North American subgroup from the Plato study. So if you remember from that slide, it was a 1.25 hazard ratio with a confidence interval of 0.93 to 1.67. So we're going to create a prior density around this estimate. I'm going to also include a non-informative prior, so weak normal, the one that has a really wide tails of normal distribution, to basically give no information to the likelihood from the prior information. And then I'm also going to include the historical optimistic prior. And so the thing that we did here that's a little bit different than the previous prior is that we chose the point estimate of the overall Plato study, but the confidence interval would have been too dense around the estimate because the trial was very large, 18,000 patients, and we're only going to get just around 1,000. And so we ended up taking the confidence interval size from the first one and surrounding it around this estimate. So it's the point estimate from the Plato study, but using the standard error from the North American subgroup. And the purpose of using this range of priors is everyone's going to come into reading our results from a different perspective. And if you have a belief that that Tycagular is not going to be helping patients compared to Clopidogrel, then you might be coming in with this one. If you're indifferent and you don't know which one's better, you might be coming in with a non-informative prior. But then if you really think Tycagular is better, you might be coming in with this optimistic prior. So it gives a range of results that combine them with our data to kind of see where it would change and kind of give a sort of a variety of outcomes. And if our posterior for all three are similar, then you have kind of a robust estimate, which can be very valuable. So where I'm at with this one, we're recruiting patients still because we had some COVID-19 delays that slowed down our numbers, but we're hoping to be wrapping up by the end of February. So we're in the process of shutting it down. So I'm hoping to submit our recruited numbers to RAMQ to get our outcome data in due time. And then we're in the process of evaluating our trial to get feedback from clinicians for the delivery of our trial, just to get an idea of how we could improve the execution for anyone who might be interested in using this kind of study design for any future results who don't have endless resources. And so I don't have much data because I don't have any outcome stuff, but I pulled this off this morning to give to you. Research nurse Nina told us that we have around 1,000 patients. We have around a mean age of 67 years old. And then we can see the distribution of what type of ACS diagnosis they had, as well as how we do have more males as we expected, and that we do have some imbalances in the people who are already being able to join our study. And that's, I think, partly due to our COVID-19 restrictions that happened in a Ticagrelor month. But at this point, the overall impact of my thesis research hopefully, does a pretty comprehensive synthesis of the literature of these three regimens. It's going to add a lot of North American evidence, randomized evidence first, and then as well as real world evidence from the observational data in a North American population. And we're hoping that this will help update or at least guide our clinical practice and maybe improve those resources that are in our current Canadian guidelines. And so with that, obviously I have to thank my supervisor for helping a lot with this, my thesis committee of Dr. Dana Curry and Sony Dondona, and obviously my research nurse, Nina, who spends her days at the hospital recruiting patients. And it's been an absolute challenge during COVID where she has to, kind of dance around a lot of obviously important medical procedures going on and restrictions. And so it's been a very tough challenge for her in the last year. And then of course, Lynn, who is, absolutely magical in helping me so far in talking about data. And I do know I'm going to be coming to bug you in due time. So again, this is the completion of my presentation. If you do have any questions, the floor is open and I will go check out the chat now.
Stephen, thank you very much. It was, I think, a very, very nice presentation that I think clearly demonstrated the scope of what you want to accomplish and the methods that you're employing. So I hope there will be a little bit of discussions, because we can always benefit from these discussions and some feedback. I have a few comments, but I will wait and let's see if there's if there's others in the audience who want to contribute. I will say that Kaberi sent a message which she apparently didn't want to necessarily send in the chat, but it is actually a very appropriate message for all of us who do any sort of administrative work, and it's actually a very encouraging message that we will end up eventually having our own McGill site, and not initially in the confines of Core, but at least on the McGill campus. The only thing I would add to that message of Kaberi's is I would sure like to see them push and get it up and running before October of this year. I think it's really an essential part of making McGill and indeed Quebec competitive again in this whole field of comparative effectiveness research, because the current barriers are such that we're just not able to play in the same game, at least not with Quebec data.
Totally agree, Jay. And actually, I did have a question for you, Stephen, at the bottom of that copy and paste.
Yeah. Okay. I saw the question afterwards. I wasn't quite sure what the question was, but
yeah,
the first part was so important.
So important.
And I think it was absolutely appropriate to stick it in the chat.
And actually, I will say to everybody who's here, if you could email me if you or your team are going to be using this center, which is the Quebec Health Administrative Data Center that we'll have on site with not only health administrative data, but other data, please let me know, because I was involved in pushing for the center, and they want to know what the volume of use is going to be. My question for you, Stephen, so, you know, I've taken to listening to JAMA clinical reviews as I try to exercise and injure my back. And the most recent one that I listened to was a discussion, I think, of the paper that I pasted the link into, but it was an observational study, multiple countries, looking at this question of Clopidogrel versus Ticagrelor. And I was just wondering if you were aware of it. And they used a lot of propensity scores, and they had a big South Korean cohort and an American cohort. And ultimately, their conclusion was that there wasn't any efficacy benefit, and actually there was more bleeding risk with. Did you see that study, and how would it maybe inform your own observational study?
Thanks for that question. I don't remember that study off the top of my head. I have to admit I've been more focused on the RCTs as of late, but it doesn't mean I have some. I don't think I have this one that you talked about. I've seen a couple observational studies pop out that I have in my reading list of to do. And I have one in circulation that is done not necessarily to look at the observation data alone, but it was the target trial framework to compare population level data in the US and try to emulate the results from that Trident and Plato study. And they did that essentially using observational data. So I think when people use observational data, you have to be careful about what question you're asking when it's relative to the population you're using. And that comes down to what the selection differences are. And so it could be that at a clinical level, there could be different results than from an RCT level. And as you said, I don't know the results, but it sounds like they didn't see any efficacy differences. And it may be because the RCTs are highly selective and don't actually represent everyone in the population. But I don't know the exact answer to that, but it will be interesting whether or not people start adhering to trying to do more observational data. And I think this is interesting, and setting it up in terms of real world evidence, but being very explicit about how you design your studies so people can try and emulate them. And it's hard to emulate RCTs in observational data. So I'm not necessarily surprised by that discrepancy by any means.
So maybe just take a look, and I didn't take a thorough look at all, but I'd have to say you're doing quite a lot. I think Jay will have you on his hands for 10 years with the scope of work that you're planning.
Well, we have to be careful because, as you point out, Kaberi, there is a lot of scope in what Stephen is taking on. That the paper from JAMA, it's Ricky Turgeon, who's out in the University of Alberta, and actually he published about a year or so ago The first study he did was looking at, I think it was Alberta data, using an Alberta database,
I have
and
that one,
saw
yes.
no effect. And which I think is actually probably what the truth is. But of course, the powers that be that fund the big RCTs and their key opinion leaders have been out in force in the cardiovascular community trying to discredit this sort of observational work. I think as Stephen says, I think, you know, we're learning more about how to do observational research well. And this idea, you know, the following the Harvard group of really framing it as an emulation of an RCT, I think is a pretty key step towards trying to limit some of the biases and that we might run into with you know, poorly done observational work. But I hadn't actually seen that paper in JAMA Internal Medicine, but I just looking at it quickly, I can see it's just a continuation of what the work he did originally with just the Alberta database.
Mm-hmm.
And if I can add quickly to that, because I think this is my presentation, Jay. The thing that I think I've noticed in terms of what I've reading and what I've been working on recently, and that the discrepancy a lot of the times between RCT data is that they always follow this intention to treat approach, even though you have exposure strategies that can vary over time. And we often don't really account for that very well, and understanding that they can impact how your results, what your results are. So this is, I think, something else that's different in a lot of the observational studies. As you notice, my nested case control design is a time varying exposure definition versus my RCT is going to be an ITT, which we kind of have been struggling with in terms of whether or not they're parsimonious in what they're asking a question about. And so sometimes we're asking different questions with these studies altogether, and it's not always whether or not one's right or wrong. They both could be right, but they're just asking different questions.
Yeah, it's the difference between the ITT and the per protocols in a different way. Very nice. Good work.
Stephen, I don't know if you see that there's a question in the chat for you from Teresa.
Oh yeah, Teresa, am I concerned with the quality of RCTs that were initially included in the meta-analysis? So I do know of some that are a bit more questionable, and I don't expect them to be kind of like, when I exclude a lower quality RCTs, they won't be in the final. So I do know some exist. How will you summarize? Are you okay? So you have two questions here. So I'm not going to. Now this is easier.
Ask you in person. Hello. Thank you for your talk.
It's nice to see your face too. So this is good.
It's rare that a PhD student would complete 2 of the studies that you're talking about, and then the third one, it's amazing. Usually, if somebody finishes 1 RCT, that's a success. I should mention that I found it troubling that you were talking throughout your talk about efficacy, and the first comment of Jay was that you were doing a comparative effectiveness study. So I really think efficacy and effectiveness really need to be clearly understood here.
That's a good point.
I have a problem with systematic reviews and meta-analyses that just put studies together without considering the real quality of the data that have gone into them. And so I would urge you to look at those studies and be sure that you know, that you're not just doing a sophisticated statistical analyses with low quality data.
Well, I agree with you, and I didn't explain it very often or very much in this presentation because, well, first of all, I find the Bayesian network meta-analysis of these 3 the least exciting. So I figured I wanted to focus on the second 2. So I do plan to account for it. I mentioned I'm going to use the ROB2. I didn't mention what my cutoff is going to be. So I do plan to exclude ones that I find to be of lower quality. I have it written down somewhere. I don't have it at hand. So
Stephen,
I do plan to.
My comment actually goes right to the basics of, for example, even the recruitment processes of these individual RCTs and the study population characteristics of the original RCT. So my comment actually goes back in history for those trials. I mean, I think Cochrane has you know, sort of covered the world now, but some of the work that they produce is garbage.
Yes.
And I think that you would do well if you would look at those individual studies. Anyway, that would be my suggestion.
I will have to get more details in the specifics of what you mean by that, I think, because I haven't really, I guess, fully understood. But I agree with you in the sense that I hope I've highlighted that I don't necessarily think that, because I think in terms of what the review outlines are, that I'll be including, for example, the Plato and the Triton as high quality studies in my review, but I've highlighted that there are issues with them. And so I don't think that on itself that you can magically believe that an RCT is, or a meta-analysis is just good on itself. You have to look in the details. And that's why I want to kind of look a little bit deeper. And I'm doing this more because I want to understand what is available and what exists from a systematic standpoint. And by no means do I think that my meta-analysis is going to be my answer to my questions.
Okay, I think a real contribution can be made with systematic reviews to expose the challenges and to expose perhaps the weaknesses in previous researchers that have combined studies when they should not have combined studies and done pooled analyses or meta-analyses. Anyway, I think that that's what I would expect about that. And yeah, and you also, just to bring up another thorny issue, you never mentioned the word ethics. And I just wondered about your RCT and ethics and how you have managed that aspect of your RCT.
In what context of ethics? In terms of
did you apply? I mean, how long did it take you to apply for ethics? Did you go through the McGill one or the hospital? Obviously, I guess you went through the hospital one. Is it just MUHC? It's not the other McGill hospitals. It's just MUHC. I mean.
So yeah, it's the REB through the MUHC in terms of the ethics boards. We applied and got approval through the research ethics boards at the MUHC. So it includes all the hospital systems there. In terms of ethics, like I think for our pragmatic trial where we get a little bit of kind of not that you should ignore ethics, but less of an ethical conundrum is that our drugs are already approved, right? And so it seems like physicians, at least if you talk to the ones at the MHC, they kind of are not sitting completely on one or the other. So there already is some sort of equipoise in the clinical practice. And we'll see in the data set, I didn't pull up the data on it, but there are prescriptions in large numbers of all three of these drugs. So they're being implemented across the board. So we did go through ethics. We went through a back and forth. We're getting complete patient signing off, even though I think we discussed that because these are approved drugs and we don't have to really decide in terms of ethics of which one they are getting exposed to. Our ethics mostly was getting their data in terms of understanding.
You answered my question because I did want you to bring up the principle of equipoise because it hasn't been, I don't know, obviously Jay is the expert on this, but I would have liked to be convinced that there was equipoise for this RCT.
So Teresa, if I can just add a little something, because it does, again, highlight another contrast between Quebec and the rest of Canada, and particularly, say, Ontario. The patients in this study, there is a complete equipoise in terms of the different DAP regimes. And that's why, you know, the Department of Cardiology, the division, has accepted to participate. But the interesting thing is that it took several months to go through ethics. People sign a 6-page consent form to participate, even though we do not, they are getting standard of care whatever month they're in. Even though there is no extra visits, there are no extra bloods that are taken, it's just nothing more than quality initiative, if you want. But they have to sign a 6-page consent form. In Ontario at McMaster, because I chair one of their internal research committees, the same research looking at 2 established drugs and at the University of Toronto gets a pass through ethics because these are accepted indications. There is no consent form. It is exactly the same pragmatic trial, different question, different drugs, but the exact same format. They do not even have to go to ethics. They get an automatic pass. It took us almost 6 months and a 6-page consent form to do the same research in our environment. You know, I'm not saying who's right or who's wrong, but I would point out that there is a lack of standardization between the provinces in terms of what's required ethically. And you can argue, you know, they're too lenient, we're too severe. I don't take a side in this, but I do point out the inconsistencies, which are enormous across the country.
Thank you for that detail, Jay. It's Stephen, it's exactly that kind of detail which most of our PhD students then only do one study. But totally understand, and it will provide a richness to your thesis to explain just the ethics component of this, which I think it would be a contribution in your lit review or the background to your RCT. Thanks.
Yes, thank you, Teresa, because you raised some important things in that. I haven't decided how to frame, for example, the Bayesian network meta-analysis or some of these other issues that could totally come up in some interesting discussions or be interesting to talk more broadly than just within our small conversations here. So thank you for those suggestions.
You're welcome.
Stephen, I think Dr. Smith has a question. If I'll be quiet and you can answer his questions.
Thanks for that very nice talk. Had a quick comment just on Jay's point. I agree that there's quite a bit of heterogeneity in terms of what's required for what seems like, you know, relatively low risk research. And I think in some ways it might be worth pursuing as a research question if we were to survey in detail and in a standardized way what the general population's perspective is on risk and benefit. It might be interesting to show that actually probably the average person would think that this sort of Ontario model is acceptable for the potential gains, because I agree it has been an impediment to research. But back to your specific projects, what was the clustering interval for your trial? How many months was it?
2 months.
So it seems sort of completely out of your control, but unfortunate if you look at the temporal dynamics of the COVID waves. Some of the sort of spikes seem to be on a 2 or 3 month time or sort of time period. I've pulled from the data warehouse a bunch of information related to COPD, acute coronary syndrome, and GI bleed, looking at the number of emergency department visits, some indices of disease severity at presentation, objective indices like troponin levels or PCO2s, and I was interested in whether, you know, the people who did show up, were they sicker at arrival, things like this. So I was curious if you had, what your thoughts are on how COVID may influence the process of this trial and potentially the results in terms of who's showing up, who's getting enrolled, who isn't showing up, who isn't getting enrolled, and whether that might introduce some sort of inequality in potential confounders, measured or unmeasured.
So that's an absolute excellent question. I admit I haven't been able to spend as much time as I'd liked based on managing multiple projects here and obviously being stuck at home the entire time during COVID and trying to organize our trial in itself because our research nurse got stopped in trying to manage when she got back up and running to collect patients. So I know I've kind of, as I've kind of like paid attention to COVID process, the kind of development, I know I've read a lot about how sometimes these kind of events can actually show a reduction in certain numbers that present. And so I haven't been able to look at the underlying data just yet because I've been trying to be, I guess, conscious that I don't want to start sifting through before I collect all my patients about making decisions on how we collect people and where we emphasize things. And I've never been against, if we do have imbalances, to adjust for those imbalances in our analyses. So that's something that has to be as a possible solution, or to provide some convincing that if you do have imbalances, you have to make some corrections for them. So I think that COVID might reduce the number of patients that will come to the hospital, because if you're doing less activities, maybe you'll have to notice your symptoms less often or not have the time to have it. But I don't know. I'm going to have to kind of wait and see and kind of investigate what the impacts of those are in terms of our population we've recruited and how different they look after this.
Yeah, I think adjusting is reasonable, but of course you can only adjust for the things that you've measured. If there are imbalances, it might be signs that there are also unmeasured confounders. And all this to say, I'm happy to share the data that I have, because to my surprise, while there's far fewer present, hospital visits or ED visits, by most simple measures, the disease severity is about the same of those who do present, and their in-hospital mortality is the same for these specific outcomes that I mentioned. So that may be a defense that people may not be so different. But anyways, thanks for your talk.
Thank you for that. And I think the problem maybe for us is that we couldn't recruit during certain sections, so we might have lost a lot of patients in certain arms. And that, I think, is one of our limitations in that. And that's why we're leading towards more talking about it, adjusting for it. But at the same time, like, we are collecting a pretty comprehensive baseline confounders. And unless there's a confounder a really strong one that exists that we don't know about already, that I feel like in cardiovascular medicine has been pretty well parsed out, that we kind of have a good idea of what is important. I'm not too worried in terms of having something in the background that's hiding away just yet. But I'd love if you have some ideas about looking into other confounders that may be of big importance here. I'd love to hear from them.
Stephen, you may have addressed this. It's Kaveri, just I'm just wondering about allocation concealment, because you're alternating, so people actually will probably guess which arm the patients are in.
It's open label.
That's, and that's always the question: would it influence treatment? If there's a belief that Tycagrelor is better, then will they treat the other patients in a different way? What are your thoughts about that?
So that's a great question. So this is an open label trial, so everyone, the physicians, the patients, they all know which drug they're on. I can definitely believe that there could be some sort of bias, cognitive bias, in terms of how patients treat themselves, or physicians treat patients differently, and even how they might recruit patients to this study. I guess the one way that we can try and answer that is that we are in our process of evaluation form. We are asking our clinicians, because we're surveying our clinicians involved, about their viewpoints on Ticagrelor and Clopidogrel in this study. So which one they think is better and what level. So we kind of have a range of viewpoints. So I don't know if that's an answer in terms of a solution. I think it's a problem that's going to exist, and we at least, I think, have, I'm assuming, outcome evaluators that probably aren't going to have strong feelings about it, so we won't try to choose an outcome to the other. And we have pretty strong outcomes. So if someone dies, we're not going to have a certain number of outcomes more in one than the other. So I don't have, I think, too strong of an issue with the open label aspect of this trial, and the fact that I think it's kind of with the equipoise. It sounds like people are pretty comfortable with either drug in our population.
Stephen, I would just add to that, for patients that are hospitalized within the MUHC, that stay in the MUHC, when they're recruited into the trial, before they're recruited, Nina actually consults with the attending physician to make sure that they are in agreement that the patient is randomized to whatever the drug of the month is. So from that aspect, for the internal patients, I don't think there's that much of a problem with following the randomization process. We also treat a lot of patients, though, with their acute coronary syndrome and then send them immediately back to their referring hospital. And in that case, they're sent back on whatever drug they've been randomized to in the month. And it is conceivable that one of the physicians in one of these other hospitals might not like the drug that has been chosen and change it. However, my personal experience is that generally these peripheral hospitals, when they get a patient back from the tertiary care with a certain prescription, particularly when there is a letter attached saying that this patient is in a research project, are unlikely to change the medication. I think the changes are mostly going to come about if they're due to side effects or whatever. But again, it's a good question, Kaberi, and something we have to be concerned about. But I think we're probably going to be okay on that level.
Interesting.
Right. Well, it's I don't know. It's after 5 o'clock. Again, I think Stephen would be more than happy offline or email to entertain any further questions and suggestions. I think Ben, I think your offer is something that we'll look at as well in terms of I have looked at another project a little bit about this question of intensity of the cases that we're receiving during COVID, but I think any insights that you might have with your data would be something we'd like to explore and you could share with us. So I'd just like to thank Stephen for a really very thorough presentation. And I thank everybody for attentively listening and for the discussion. So,
yes, thank you,
Stephen,
everyone.
thanks again, as usual.